I'm here today to talk to you about some of the technological advances to revolutionise therapeutic drug monitoring. And that's both in the areas of sampling, where we're looking at reducing the volumes of sampling, but also some bioanalytics. So some of the sampling challenges for TDM fall to both the patient but also the laboratory. Certainly for the patient, some patients can experience difficulty just in the volume of sample that's required. One of our speakers earlier talked about Amanda Gree was talking about the volume required for TDM amongst the other clinical samples that you'd be taking. And this can be a real burden for neonatal or paediatric patients. Also repeat sampling, so if your TDM is, again, there's that burden from continued sampling. Some patients have challenging phlebotomies, and that can be difficult to take a venous sample. Certainly patients who are outside the hospital environment, so out in the community, as Kate was talking about, or outpatient clinics, there can be challenges in getting to the clinic and then having a sample taken, particularly if people are working, and then patients in remote and rural areas, and this is where they can have a challenge in getting into clinics. Certainly in Queensland, we've got patients who might be 4 hours out of Gympie, and to perform TDM they need to travel in 4 hours just to give a sample before returning home, and then there can be a delay before the sample, before they then need to go and see their GP or specialist regarding the result. And the other aspect of sampling is the resource constraints. So currently it requires highly specialised equipment for measuring drug concentrations. These are usually in a centralised laboratory. In Queensland, the size of it is, that is, Pathology Queensland have samples from Thursday Island, I think it's 4,000 kilometres from Brisbane, and those samples need to come into Brisbane for the TDM. So that's another aspect, and I'll talk a little bit more about that in the bioanalytic area. So one of the areas of my research is looking at microsampling and how this can be of benefit to patients. So I thought I'd walk through with you some of the different types of microsampling. What you can see here is dry blood spots. These have been in use since the 1960s for neonatal screening. So in neonatal screening, what they're looking at is a yes/no answer for a problem, but we're looking at using it where we measure a drug concentration. And I'll talk to you more further about that. The next one is a capillary microsample. So these are used for point of care testing in paediatrics, and we're looking at really repurposing this. The nurses in the clinic are very used to taking a sample from a heel prick into one of these tubes. They take approximately 50 microliters of sample, and what we're doing is, instead of them just then applying that into a point of care gas machine, is that we actually seal the bottom of it and centrifuge, and from that we can harvest the plasma, and we can use that plasma for an analysis of a drug concentration. The alternative, if the patient was remote, you could also apply that to a card and produce a dry plasma spot. I've got a short video, if I can get this working. So this is a device called a volumetric absorptive microsampler. And in this video, what you can see is a drop of blood is collected onto a hydrophilic tip, and it contains 10 microliters of sample. So an advantage there for me, where I'm trying to measure drug concentrations, is I have a known volume, and that makes my life a lot easier. I've got another video for you, which is just here, and this is where I'm collecting a sample. There's a lot of self-sampling going on here, where I'm collecting a sample into a microsampling wing, and this is a microfluidic device, and as you can see, we just collect blood from a finger or heel prick. It moves through a fine microchannel, which you can see in this end picture here. It's centrifuged, and then the top part of the microfluidic device can be broken into parts. You can collect there's a duplicate analysis here of 2.5 microlitres of plasma, sorry, 2.8 microlitres, and then they have what they call the lucky zone, which is here. And if that's filled with plasma, you'd be able to obtain a third sample at 2.5 microlitres. So I guess what you can see is there's a lot of different types of microsamples. And what I would put to you is that there's no perfect microsample. It depends on your patient, depends on what you're trying to measure and what you need. So if your patient is in a hospital, you can still have an advantage of a reduced blood volume, but you could keep the sample as a liquid plasma sample. There's no need to dry it. You can still harvest it using centrifugation. If your patient's in a rural or a remote region, you might prefer to move to a dried sample so that the sample can potentially be mailed in. Okay, and so I put this to add a little human element to my research. This is a photo that's similar to a neonate that we were sampling last Friday. It was a 24-week-old. And I think that, to me, is the importance of microsampling, is there's patients who can't provide large samples. And this was a patient in which we were able to collect 20 microlitres of sample and get a valid result from. And I think that's really important for that aspect of of our patients. The patient on Friday was in our NICU here at the Royal Brisbane Hospital, which is called the Grantley Stable NICU, named after Dr. Grantley Stable. And he made the comment that neonates have equal rights to the same degree of care as the rest of the community. And I think that's where I can see microsampling being of real benefit to patients. So some of the advantages of microsampling to facilitate therapeutic drug monitoring. Talked a lot around the reduced blood volumes, but there's also simplification of obtaining a sample. Dried blood spots can be cheap and simple. Finger prick sampling is quite a simple process. And there's also the potential for self-sampling, as I've demonstrated. There can also be simplification in processing. That's in the case where a concentration can be measured directly. There can also be a reduced cost in shipping. If you haven't got a liquid sample, you don't need to freeze it, store it on dry ice during transport if it's dried. You may also find that you've got added stability during storage, and I'll talk a little bit further. Actually, so we've done testing of vancomycin on the VAMS device, which has been dried, and we've found that it's stable at room temperature for over 100 days. And dried blood samples also have the potential to reduce the biohazard risk. And there's just a couple of papers there talking around therapeutic drug monitoring with the use of microsampling. So if you want to implement microsampling into the clinical environment, there's a few aspects that you need to consider. As Jan Willem mentioned, there's the analytical side, being confident that your measurement of concentration is reliable and accurate. And this is some guidance that is provided by the FDA and the EMA, where they have a prescriptive type of analysis and a testing that needs to be performed to ensure that your sample is, the integrity of your sample is retained. And just very recently, they started to talk about bridging data, and this is where the FDA is starting to move towards acceptance of alternative technologies in terms of dried blood spots. So after you've performed the laboratory validation, we then move into a clinical bridging study. And this is where we're comparing in patients microsampling to the gold standard of plasma sampling. And this is where we're understanding the difference where we might have traditionally collected an arteriovenous sample and we're now collecting a peripheral sample. The FDA's guidance is that it should be suitable for intended purpose. And so there's some discussion within the scientific literature about what is an appropriate study result, what is an appropriate result from a bridging study. One one of the tests that's being performed is what is usually called an incurred sample reanalysis, and we're applying that test to our bridging study data, and which, when you collect the microsample and the traditional sample, the two techniques have to be less than 20% different between the mean of them in 67% of the samples. This can be visually represented on a Bland-Altman plot, and I'll walk you through some of those in my talk. And one of the other aspects of our research, which I think is important, is we're also looking at patient acceptability, and we're including qualitative research in our studies so we can get an understanding. It's easy for me to say finger prick sampling is less invasive, but actually there is a pain associated. And so it's understanding that for the patient and what do they prefer. We recently published, so there's a study here by Jan Willem where he was looking at the use of therapeutic drug monitoring, and he included a qualitative aspect to his study. And we recently published some results from renal transplant patients here at the Royal Brisbane Hospital. They were found to have a high tolerance to finger prick and venipuncture. And in some ways we need to ask, what's the patient used to? The results compared to a study, say, with healthy participants, where they found venipuncture provided moderate pain compared to finger prick, so they found the pain more from venipuncture. A renal transplant patient is quite used to venipuncture. This is part of their clinical care. So that's, the difference in there that we see could be just in the different patients. We found that in the renal transplant patients, 85% of them preferred BAMS and DBS to venipuncture, and 95% of them were interested in self-sampling. The burden associated with venipuncture, the patients found that it usually took, in 43% of the patients, it usually took more than 1 hour to provide a sample. And they made comments about the challenges for a patient in late opening clinics where they would be already meant to be starting their working day, but they weren't able to because they had to go and sample. So we're just keeping an eye on this area of acceptability to the patient and whether or not this is of benefit to them. A little bit around the dry blood spots. So this was, a lot of people felt that dry blood spots were really going to revolutionise sampling, and there's still plenty of data to suggest this is a great way forward. But one of the areas they found was the challenges of sampling, in that if you were to sample a blood spot, what you would find is you can find different concentrations at different places in the spot. And you can have a concentrating effect around the outside, or sometimes a concentrating effect at the inside of the sample. And that means if you're subsampling, you can get a different concentration depending on where you are in the spot. This was also a study of dry blood spots from patients in 4 countries, and 46% of the samples were rejected. 34% were rejected due to incorrect blood spot collection. This was where they were able to tell that the finger or capillary touched the card. That was usually by the shape of the spot. It wasn't a perfect ring, or they could see the marks from the capillary. And 12% were rejected due to inappropriate drying. So that's, a real challenge, but it's also around the area of training and people understanding about sample collection. This is a study also Jan Willem has been involved in, and this is the therapeutic drug monitoring of antifungals. What you can see here is he performed his laboratory-based validation, was able to get excellent precision, less than 14% accuracy between 91 and 105%, and it was stable at a variety of temperatures for 12 days. He then took it into a clinical validation, 28 patients, he collected 30 samples. And he also included this element of a qualitative research, so asking for patient preference, and found that the patients did prefer, on a pain scale, preferred the DBS. The DBS generally took 4.4 minutes for the samples to be collected, and 100% of respondents found that was an easy way to collect samples. This is the results of a study that we've been running here, where we compare capillary microsampling to microsampling wings for measuring vancomycin concentrations. And this is one of the Bland-Altman plots I threatened to show you. So what you can see here, I've got lines at 20% and minus 20%. And this is the acceptability criteria within an incurred sample reanalysis. So you need 67% of your samples to fall within that. So what you can see with the capillary sample, we had a bias of 0.2%, with the microsampling wing slightly higher at minus 3.7. But what you can also see is across a concentration range, there's no obvious, although the variability is fairly similar across the entire range. So both of these look like they'd be useful going forwards. We also have the VQuin PK study, which I'm doing with Andrew Burke. And this is in 60 household contacts of patients with MDR tuberculosis in 5 clinics throughout Vietnam. And here we used VAMs to collect samples for a PK sub-study. And in 28 of 30 paired samples, we found that 93% of the samples were within 20% of the mean. So it does meet that criteria. Again, another Bland-Altman for you. You can see there that the data sitting within the 20% criteria. And you can also see I've got an upper limit of agreement and a lower limit of agreement. That's your 95% confidence intervals of data. Our laboratory-based methodology found that it was stable 11 months at room temperature and 6 hours at 50 degrees, which we tested because we were transporting from Vietnam to Brisbane and we weren't sure if samples would get stuck on the tarmac or, incur those sorts of temperatures. But as far as bioanalytics goes, one of the areas for TDM is the measurement of concentrations bedside. And to be able to do this would be a game changer, and I think people would be greatly interested in seeing this being able to be done. And I've got 2 examples here of groups who are trying to bring this into the clinic. This is a group in the University of Southampton, which I'm working with and enjoying collaborating with. And then what they're looking at using is refractive index, so Fourier transform infrared, to measure blood concentrations bedside. And it's quite large on that big screen, but it's actually only a small device, about 20 to 30 centimetres, so it could sit bedside. And what they found is that the refractive index across the full scan is dominated by water. Whole blood has features of protein content, but when you remove these and you look at a particular area of wavelength, then across a concentration range, you can build a calibration curve. And in this example, they're measuring, I haven't included that, I think it's amoxicillin. Sorry, guys. And another study is one here, and this is a German group who are looking at clinical on-site monitoring using beta-lactam antibiotics. And what they're doing is they've developed a microfluidic chip where a sample is, I need my glasses, sorry. So a blood sample is placed, there's an immobilization area which is based on binding with to penicillin binding protein. And this is a competitive process between the analyte and ampicillin biotin. And what happens is when you apply the enzyme glucose oxidase, it binds specifically to ampicillin biotin. And when you produce when you apply glucose, hydrogen peroxide is produced. And so if you're measuring, they then measure using an electrochemical cell the amount of hydrogen peroxide. And because it's a competitive binding, the amount of hydrogen peroxide is inversely proportional to the analyte concentration. So, and as you can see, this is the sort of system which could sit bedside. And then I was just handed a paper which has just been published that both Jason and Jeff have been involved in, where they're talking about on-site therapeutic drug monitoring. And it mentions half a dozen types of bioanalytics that are going forward in the hope to measure, to be able to do TDM. And that's all. I just wanted to say thank you very much to all the teams that are helping. And if you've got any questions, feel free to jump on chat, or you can find me on Twitter. Okay, you can.
I might ask you a question, Susie. So in terms of what antibiotics are amenable to microsampling, so can you do potentially microsampling on any antibiotic, or are there certain ones which have characteristics which prohibit that in terms of stability and things like that?
Yeah. So that would be exactly it. So if if you can't find stability for your microsample, then I think there'd be challenges around it. But that's why I don't believe there's a right microsample, because even an unstable compound, you could potentially collect a liquid plasma sample and store it at -80 degrees. So yeah, it all comes down to the type of microsample and the patient and what the result is that you're after.
Thanks, Susie. So I must say, when we did our study in Vietnam with levofloxacin, it was amazing how well received it was. It's very easy to teach people, like Susie taught me, and I had to teach other people in another country through an interpreter, but it was really, and the results we've got back here subsequently validated that process, so it's really good. I'll just see if there's any questions online. Quick question, is microsampling compatible only with certain devices? So I'm not sure whether that's referring to...
Hang on a second. Oh,
sorry.
Okay. When the patient provides a dry blood sample, do they provide just one spot or blob multiple spots? Yep. Thanks for the question. So that's so for dry blood samples, the recommended way of collecting a sample is to allow the drop to fully fall from the finger and land on the card. From the one finger prick, you can collect multiple samples. So they could collect 3, 4 spots, and these could be used to measure the concentration levels. As for the mean concentration, I think what you would collect concentrations from the 3 spots if you've collected 3, and then you'd probably calculate a precision around that and present a mean. Okay. So quick question, is microsampling compatible with certain devices? I'm not actually sure, Jennifer, what you're asking there. Do you? Okay, but if you want to find me on Twitter, I'm there. Okay.
Thanks. Thanks. That's great.